This year's Darwin Festival Sigma Xi speaker is Dr. Erica Polleys. She's an assistant professor of biology and biotechnology at Endicott College. She completed a Bachelor of Arts in Biology at Suffolk University and a PhD from Baylor College of Medicine in Houston, Texas, where she uncovered important molecular pathways in the cellular response to DNA damage. She continued her studies of DNA damage and repair in a postdoctoral fellowship at Tufts University, where she focused on learning which steps in the process of homologous recombination drive CAG repeat instability. Dr. Polleys' work has led to multiple important publications, and her CAG repeat expansion work has important implications for understanding genetic mechanisms leading to Huntington's disease. So we're very honored to welcome Dr. Erica Polleys for her talk, and you can see the title.
So nice to be here today. I'm so excited to talk to you. So I study DNA damage and its consequences. And DNA damage can be initiated by lots of different events. So we could have endogenous sources, things like replication errors or reactive oxygen species, things like hydrogen peroxide, but also DNA secondary structures, which I'll talk a lot more about. And it could also be due to exogenous sources, so things outside of our cells. This could be things like ionizing radiation, pretty bad. Chemotherapeutics, a lot of them induce DNA damage. And then our favorite new biotechnology in the, nowadays, CRISPR-Cas9 genome editing induces a double-strand break. And so all of these events in our lives are coming together to maybe start DNA damage, and then we have to do repair. And if cells don't repair their DNA perfectly, mutations can happen. And so then we have mutations, maybe more DNA damage, and more mutations. And eventually we get to the big C, cancer. And really, that is a lot of what my research is focused on. So I want to take a step back and kind of think more broadly. Let's try to make relationships between something that we all know and experience and then relate it to molecular mechanisms, which I feel like as a student are really hard to imagine. So, what if the cell was a house? And within that house, you have all of the rooms, all of the places and systems in place for you to have a perfect set of homeostatic things. You have heat and hot water and a nice cozy bed to sleep in. All of these important things. But what if you hire a bad contractor and we can't fix some of the things that are going wrong in our cells? Well, say we have DNA damage that can't be repaired. One option is to undergo programmed cell death or apoptosis. And that's actually a good thing in the instance of cancer because it at least means that something bad has been removed from the system. But a lot of times the cell will just continue to grow and divide because it's accumulated mutations that make it more favorable to do that. And this could eventually lead to cancer and really late stage problems in terms of metastasis, et cetera. And cancer really is a disease of mutation. So I study DNA damage and DNA replication, and these are both implicated in cancer outcomes, as well as generally something called genomic instability, which is a funny way of saying a lot of stuff is wrong in our cells in terms of the DNA content. So if cancer is a disease of mutation, and I'm interested in DNA damage, then maybe what could happen is we have a DNA damage event, and these damage sites are normally pretty well detected by the cell. And this results in lots and lots of signal transduction that results in accurate DNA repair, cell cycle arrest, so we stop things, and we say, hold on, we're gonna fix this, and then we'll start doing this again. or shuttling it into apoptosis. And if any of those systems fail, then we're going to start to get situations where we have mutations and cell division that is kind of uncontrolled. And so DNA repair pathways protect the genome from mutation. And I love DNA, so I'm going to tell you all about it for a minute. So we know as biologists that DNA is a double helix. We learned this in the 1940s when Watson, Crick, and Franklin all were kind of running up against trying to figure out the structure of the DNA molecule. We have 4 DNA bases. We have A's that pair with T's and G's that pair with C's. And this gives this inherent matched pair to the DNA strands that run in this anti-parallel direction. The double helix is incredibly strong and stable, and this is due to a lot of weak collective interaction. And because of those weak collective interactions, we are able to maintain genome fidelity through replication. So each round of replication, we can separate those strands, we can do DNA synthesis, but also do nothing. It can just be there and exist in a stable form. And we, humans, are something called eukaryotes, which means that we have a true nucleus, and our chromosomes, our DNA content, is organized into these linear X-like structures when they've been replicated. And these chromosomes are where all of the genes in your body are located, regardless of whether or not they are being transcribed and translated into proteins. Now, most humans have 46 chromosomes, and they represent 23 pairs. So we have 22 autosomal chromosomes. These exist whether or not you are biologically male or female. And then we have the 1 pair of sex chromosomes, which are that determinant. So we have someone who is XX, and they would be biologically female. Then we have someone who is XY, who would be biologically male. And you can kind of see in this colorimetric picture all of the different pairs. They all have a pretty color, and they all go together in this perfect side-by-side comparison. But cancer genomes are way different. So they're full of mutational changes. So here we have 59 chromosomes in a colorectal cancer cell. So now we can say, all right, well, we're supposed to have 46. Now we have 59. So we've gained chromosomes, and you can start to see some, but it's not evenly distributed. You know, you have something like chromosome 2 that now has 3 copies of it, whereas chromosome 5, you only have 1. So there's been this random shuffling event. And now, because we have these colors, you can also start to see that some of these chromosomes aren't fully what they think they are. They're a little bit of chromosome 1 and 2, for example. They've had some sort of chromosomal rearrangement. And then what is not pictured here, because it's slightly beyond the threshold of detection within a karyotype like this, is small single base pair changes that have led to events that eventually lead to this really problematic outcome. And so the source of deletions and translocations, like in that karyotype on the other slide, is usually a chromosome breakage event that hasn't been accurately repaired. So, I'm a first-generation college student, and my dad grew up in the greater Boston area, right near Winchester, and he eventually became a plumber. And so my entire scientific career has been trying to translate very complicated topics into things that my dad would understand. And so I think of DNA as cellular plumbing. It's really important, really essential. We feel real bad if it's not working. And there are kinds of issues with plumbing that we can kind of relate back to DNA and DNA damage. So say first you have a blockage in your water pipe. This to me is a little bit like DNA replication, which can sometimes stall, so slow down, but you can pass it usually. But the other outcome is that you have a cracked pipe, right? We've had a really cold winter, and I'm sure some of you have had situations in your homes recently where your pipes have frozen and they've cracked, and you've had to repair them. And that is a real problem. So if we think of the kind of analogy towards plumbing, say we have this problem that occurs during replication. Something is wrong. If you have a blocked pipe, you have to take care of it. But sometimes you need a professional. But most of the time you can handle this on your own with just some fancy chemicals. On the other hand, if you have a problem that occurs during a double-strand break, your pipe is broken, that can be pretty catastrophic. Do not recommend. It is expensive, it's time-consuming to fix. That's true for plumbing, and it's true for your cells too. It impacts things locally, meaning that one cell is a problem, your one house is a problem, but also globally. So what if your water main breaks? Then a whole set of systems are failing. And that's true for lots of DNA damage and lots of cells. And most importantly, this is not a solo job that you can usually fix on your own. Now, to go back to DNA damage and repair, to step away from the metaphor, there are lots of common causes to DNA damage. So you might have replication stress. That could lead to an oops in terms of adding a DNA base where it should be. So this is called a base mismatch. Then we have single-stranded breaks that are caused often by oxygen radicals, chemotherapeutics, and ionizing radiation. This is where one strand of the DNA breaks. And then double-strand and intrastrand crosslinks are mostly caused by ionizing radiation, chemotherapies, and CRISPR-Cas9. And then things like UV light will cause really bulky DNA adducts. And this means that now the DNA is distorted and has to be repaired. Now, I am interested in DNA repair because it turns, and more specifically double-strand break repair, because it turns out that inherited mutations in proteins related to double-strand break repair pathways increase cancer risk. Many of you may have heard of the genes BRCA1 and BRCA2. They're known as the breast cancer predisposition genes, and many women and men are born with mutations in either one of these genes, and over their span of life have a 4 to 5-fold increase in cancer risk relative to their age-matched non-mutant control individuals. BRCA1 and BRCA2 are essential for homologous recombination, and this pathway is a complex set of steps that is there to repair double-strand breaks. So let's go back to my house analogy, where we have our house, we have our plan, right? We have these blueprints that tell us all where all of the pipes and wires, et cetera, are in our house. And we have all kinds of different proteins that come in to repair our double-strand break accurately. So this is a little bit like our contractor. And this one is mine, my dad. And so now I'm going to step aside and give you a little primer on this DNA repair pathway, of which I am so passionate about. And this is homologous recombination. HR is kind of a 5-step process that involves anywhere between 5 and 20 proteins. It's complicated, but we can break it down into kind of unique steps. So first we have the double-strand break. And immediately, like within seconds of this break happening, we have proteins on the scene of the at the scene of the crime, as it were, to bind the DNA end and kind of tether it back together, kind of hopefully like trying to bring it back together, maybe make it easier to repair later. This is where BRCA1 really comes in. Then, after we have end tethering, we have a process called resection. This is where exonucleases, kind of like these little Pac-Mans, chew away DNA bases so that one strand of the DNA is now single-stranded. DNA does not like to be single-stranded. It needs to be protected. And so then we have these proteins called recombinases that bind that single-stranded DNA and protect it and get it ready for its next step. And this is where BRCA2, the other breast cancer predisposition gene, comes into play. Then, once it's prepped, it has all of these proteins bound onto the single-stranded DNA. It finds a homologous template. Now, what is a homologous template? It could be anything from its sister chromatid, so the most recently replicated chromosome. It could be a homologous chromosome, so one of its matched pairs, or some other site that just happens to share some sequence similarity. And so it will invade this sequence and then copy, base for base, what it was missing. And then finally, we have this final process where the DNA that was invaded into the homologous template rejoins its original broken end. There's some base pairing that happens again. And then something called gap filling takes place. And this is where DNA synthesis occurs to fill in any remaining single-stranded regions of the genome. Because DNA does not like to be single-stranded. It was perfectly designed to be double-stranded. And so I want to have you keep this in your brains because we're going to circle back. So it turns out that our genomes are wildly complicated. And there are regions in our genome that are particularly prone to sites of genome fragility and instability. So within all of us, there are sequences in the genome called common fragile sites that, when experiencing replication stress, can cause DNA damage. And then, which is pictured over in the purple picture. And then we also have the telomere, which is the ends of linear chromosomes, that also are regions of high chromosome instability and fragility. And I spent time in both my graduate and postdoctoral work studying both telomeres and these fragile regions in the genome. And they are somewhat linked, actually, these telomeres and fragile regions. So as I kind of mentioned earlier, we have this expectation, this ideal picture in our brains of what DNA looks like. It is this linear kind of twisting molecule. And we call this Watson and Crick structure, that's pictured here, B-form DNA. And that's our expectation. But the reality is so much more complex. So actually the DNA isn't always so linear. There are these secondary structures that can arise that cause problems to our genome. So we have things like triplex DNAs, G quadruplexes, single-stranded DNA loops that are caused by RNA and DNA hybridizing together, cruciform structures, and then zDNA. And all of these can influence different cellular activities. And when they become too much of a barrier to those different cellular activities, this can lead to onset or progression of cancer because the repair or negotiation of them can be mutagenic. And so really, a lot of my scientific question has been, for the last decade, has been, what if that broken pipe in my house has to be negotiated through all of these weird twists and turns. And that's what these hairpins, cruciform structures, G quadruplexes are to our genome. They are something to overcome. During my postdoctoral work, I worked at Tufts University under the mentorship of the great and good Catherine Freudenreich. She is a pioneer in understanding how these repetitive regions of the genome have to be negotiated during replication and repair. And she really focused on something called a trinucleotide repeat, and more specifically this CAG repeat. So trinucleotide repeats are where you have 3 bases repeated, one right after the other. So CAG, CAG, and CAG. Now, in terms of, so that's at the DNA level, and then it gets transcribed into RNA, and then translated into a protein if it's within a gene body. And in the case of Huntington's disease, it is found within the gene body of the protein huntingtinin. And when it gets translated, now you see this one repeating amino acid over and over again. So often you'll hear this disease called a polyglutamate disease, and that's because there's lots of glutamate amino acids in the protein structure. And these trinucleotide repeat expansions are really dynamic mutations. So here we have the huntingtinin gene, and within that kind of first part of the gene body, we have these repetitive DNA sequences. Now, for the most part, Huntington's disease is an incredibly rare disorder. And so we as individuals mostly exist below this problematic threshold. So we have less than 35 repeats in our Huntington gene. And that's cool. We're gonna live long and healthy lives. Then there are some individuals, more rarely, who maybe either over the course of their lifetime or through inherited risk start to acquire something in the gray area, something a little bit in the middle. Maybe you have 40 CAG repeats. And that person may experience maybe some late stage dementia, Alzheimer's-like feelings, spinocerebellar ataxia, so loss of coordination between your brain and your body. And then there are people who have greater than anywhere between 40 to 120 CAG repeats in their Huntington gene. And these people have full-blown Huntington's disease. And usually the more repeats you have, the earlier onset in your life you may experience neurodegeneration. Huntington's disease is particularly devastating, and one reason why it has kind of been pervasive in our society and culture is because it is autosomal dominant, which means if you have one of these expanded alleles, you will absolutely experience the phenotype associated with Huntington's disease. But often the onset now is late enough that families have already procreated into the next generation, carrying the mutations forward. And so this also leads to the fact that these are very dynamic mutations. They're also rare. Huntington's disease is rare, but there is a much broader context that Huntington's disease fits into. There are over 30 of these repeat expansion disorders that have been found over the last 40, 50 years, and more are still being uncovered. As recently as the kind of 2020s, we have discovered new repeat expansion disorders, including this disease called Canvas, which has a repeat that is AAGGG. And part of the reason why we are now experiencing this kind of age of knowledge relating to these repeat expansion disorders is because now we have whole genome sequencing at our disposal. And so the wealth of information about our genomes is now ever more accessible. And since these are rare disorders, the more genomes we have, the more power we have to discover things like this. Now, most of my work has focused on this expanded CAG repeat. Long tracts of CAG repeats can form hairpin loops. They can impede DNA replication and repair. They are prone to breakage, which I'm going to term collectively fragility. And these fragile events can lead to genomic rearrangements, and that can lead to cancer. And problems that occur at the CAG repeat can trigger DNA damage and DNA repair pathways. So, say we have this long CAG repeat, and it forms this hairpin in our genome. And then one option is that over cell division, over repetitive cycles of replication and division, we have an expansion event. This adds repeat units. So if we're thinking of the Huntington's disease example, now we have more repeats and maybe onset. We could also have contraction events. This is attractive from a gene therapy perspective. If we can think of a way to contract the repeat, then we could maybe make this go away, which is nice. We also have the possibility for further mutation, one that now destroys the CAG repeat. It's not repetitive anymore. That's okay, actually, probably. Depends on where in the genome it is. And then we have breakage as our last possibility. And expansion, it turns out, makes it worse because it can lead to further breakage events. So let's think of this repetitive cycle. We have an expansion event that causes this hairpin to form, which means that the next round of replication has a harder time getting through it, which leads to a breakage event, which then leads to a repair event that increases the expansion event. So it's this ever-winding cycle of terror. And previous work from Catherine's lab has shown that this DNA repair pathway, homologous recombination, leads to repeat expansions. So here on this graph, we're looking at percent expansions. And in wild type cells, ones that haven't had any genetic modification to them, the expansion frequency is pretty low. But then in the light blue bars, when we delete genes related to DNA processes, the expansion frequency goes up. So that's interesting. And then when we delete my favorite protein, RAD51, which is essential for homologous recombination, all of these expansions now go away. And so that to us said, well, homologous recombination is pretty mutagenic. And this is where a lot of my time has been spent for the last decade. And so really, a lot of my scientific questions have been, could the gap-filling step of homologous recombination be a source of mutation? So see, now I've circled back. And why do I think that this step could be particularly mutagenic? Well, it uses polymerases, which add new DNA bases. And famously, polymerases are terrible at their job. They're so bad at it. They need to be handheld constantly by lots of different proteins that make them processive and do their work. It also turns out that fill-in steps are pretty conserved, that there are lots of different DNA repair pathways, and almost all of them have a step like this, which means that, yes, while I am interested in this one particular DNA repair pathway, that I can also maybe make the leap to others as well. And a lot of these pathways have been also implicated in causing repeat expansion disorders. So I would say I am a molecular biologist and a geneticist by trade. They're my favorite things. This is my home. This is where I like to play. And I developed a genetic assay to specifically look at repeat fragility and instability during this one particular step of homologous recombination. So how did I do that? Well, I used something called budding yeast, or Saccharomyces cerevisiae, as a model to study DNA repair. And budding yeast is the perfect model for me. It's so easy to use. It's a single-celled eukaryote, so lots like us. Our cells are very similar to yeast cells, at least when it comes to chromosome structure and compartmentalization. And they're easy to grow, they're cheap, and anyone can work with them. They can exist as a haploid or a diploid, so they can have one whole set of chromosomes or two. And this is powerful because if we're working in haploid organisms, we only have to delete one gene instead of two. It's half as much work. Then, importantly, because I've been funded by the American Cancer Society and the NIH over the course of my career, it turns out that many genes in humans have conserved similar proteins in yeast. And this means that things that I uncover as genetically important in my yeast cells can be translated into human cell work. The genome is well characterized, and it's really easy to modify genes. Well before CRISPR-Cas9 has been modifying and deleting genes, the yeast community have been deleting them since the 70s. And it's super easy to design genetic assays that allow me to study one particular event. And yeast can be used for genetics, biochemistry, and molecular and cellular biology with great success. So I had a collaborator named Jim Haber, who is a well-established professor at Brandeis University. He's like the father of DNA damage and repair and a lot of yeast genetic stuff. And he was very kind and gave Catherine and I this genetic assay that he and his lab members had built, wherein there is an inducible double-strand break that is very simply regulated by the switch of a sugar. So yeast typically like to grow on glucose. And if we shift that to galactose, this induces expression of this nuclease, which creates one single double-strand break that is right in the middle of this genetic assay. Then what happens is all of the steps of HR that I just talked to you about. First we have resection, so making that DNA single-stranded. Then we have an annealing event where two similar regions of the genome come together. And then finally, my favorite step, the gap filling step. And this is where then polymerases synthesize through, create a double-stranded DNA molecule again. Now, what I did, how I made it special and for me, is what I inserted on the other side of the double-strand break, and that was my CAG or CTG repeat. With the idea that when the cell is trying to restore this double-stranded break and restore the DNA to being double-stranded again, it would have to synthesize through my repeat, and then I could look at changes in stability and fragility. And I designed this assay so that there were 3 different templates to consider. One where C's, T's, and G's were the template that the polymerase had to synthesize through. One where there are C's, A's, and G's. And then finally, a scrambled control. And this has C's, T's, and G's, but they're not repetitive, and they're not predicted to form secondary structures. The assay itself is pretty simple. I take a colony that grows on a rich media plate. I confirm that it actually has a repeat tract in it. And then I grow them overnight. Then I plate them on my control condition, which is a rich media called YPD. And this is where no break is going to happen. This is just instability due to life. And then I plate them on a galactose plate, and this induces my double-strand break, and this is my experimental condition. This is where gap filling is going to occur. And then the later analysis is also pretty simple. because I'm just going to look at percent viability, which is number of colonies that survive on my experimental plate divided by the number of colonies that survive on the glucose plate. And then I can also do PCR to look at changes in repeat length. And I was very early on into my postdoc. I want to say this was within 6 months. And I had made this strain, which was shocking at how easy it was, frankly. And I immediately saw that one orientation had this 4 to 5-fold decrease in viability. So if the no repeat and my scrambled control strain had roughly 80% viability, then my CTG template went down to 20%. So cells are dying. That's weird. And it's not true for the CAG template, the other repetitive sequence. And so this really became the genesis of all of my postdoctoral work. Because sometimes you find something cool and you just gotta figure it out. So I had a very, very talented, amazing student named Isabella, who did a senior thesis with me while I was at Tufts. And we really wanted to investigate what could explain that loss in viability and the decrease in repair product formation in this CTG template strain. And we noodled over it for a while, as one does in the lab. And we thought, okay, well, option one. Option one is where they repair their double-strand break as we expect. And this leads to cell survival. And maybe that's our 20%. But what if another break occurred and it happened to be at the CTG repeat, that repetitive sequence that has high fragility? Well, it turns out genomes are complicated, and my genetic assay was maybe not the best positioned in the genome, because it turns out that right after this double-strand break is an essential gene. Essential genes are required for cell survival. And so if there were a break that occurred at the repeat after I had induced my intended double-strand break, and the genome didn't accurately repair both of them, then we would lose this essential gene and the cells would arrest and die. Now, that's a great hypothesis. I named it the 2-break hypothesis, and so it must be true, right? Well, you always have to prove that. And so what Isabella did is she made a plasmid that had our essential gene on it and put it into our yeast and looked to see if now, if we gave back that essential gene, if we could start to capture these 2-break events. And sure enough, when we added back NFS1, we saw this survival and colony formation. And so it does seem like on paper, there is this double-strand break that we induce and a second break that occurs after the first. And when we looked at these colony plates, we were kind of surprised. We have these big fat colonies. These were the ones that we thought would have repaired as expected. They were growing super, super well. And then we had much smaller colonies. And we thought, okay, well, maybe those small colonies are ones that repaired weirdly. And they maybe don't have as much cell ability to grow well in rich media because they've lost this essential gene, and we've kind of given them back some of it, but not enough of it. And they've lost several other genes as well. And so maybe that's why they're not growing super well. We were interested in, so this was pretty good confirmatory evidence that our hypothesis was heading in the right direction. And we really wanted to see whether or not we could actually look at the breaks happening in real time. And to do that, we turned to a very classic DNA analysis method called Southern blotting. This is where you isolate genomic DNA from cells, doesn't matter what kind, mine were yeast. Then you often digest them with restriction endonucleases that will recognize regions in the genome that you're interested in. Then you run it out on a regular electrophoresis gel, which as biologists here at Salem State, I am willing to bet you have done in your lab classes. Then we transfer that DNA onto a charged membrane, and we probe it with sequences that are of interest to us as researchers using a radiolabel detection method, so radioactivity. And strikingly, when I started to do these southern blots, I was struck by the fact that I could actually see the break happen. So let me walk you through this. So in this experiment, I'm growing yeast where at hour 0, I add galactose, inducing my break. And then over time, every 2 hours, I take some of my sample for later DNA analysis. And in the scrambled template, what you can see is that there are these kind of 2 bands that persist over time, which was expected based off of my size calculations. But then in my CTG template strain, I saw this other smaller band that migrated further in the gel. And we were like, I'm pretty sure by the math of it all that this is my broken chromosome. And that was really, really cool to find. It was real physical evidence that not only was a second break happening, that I could see it, but that it was directly after I made the initial double-strand break. So it seems like we initiate a double-strand break, this resection process happens, making it single-stranded, and that now means that the genome is unprotected in this way that allows for another break to occur. Now, in terms of repeat stability, not only does the CTG orientation strain have this decreased viability because there's this other break, it's also wildly unstable. So in terms of looking at contraction frequency, in our no break condition, there is roughly 50% rate of contraction, and that goes up to almost 80% in my break condition. And this is not true at all for my CAG orientation strain. So again, we're at template is very important for repair outcomes and genome stability more generally. Now, as I mentioned, I love genetics. And I'm really interested in understanding what genetic factors could make this better or worse. Can we modulate the system? Can we play around and see what factors are actually doing the work here? And so we took what I call a candidate gene approach, which is where I know roughly what genes are important for this process, and I'm going to delete them kind of sequentially to see which ones kind of stick or are of interest. And we settled on this protein called RAD9. It has a human ortholog called 53BP1, which is a tumor suppressor gene. It has roles in activating the DNA damage checkpoint, which kind of stops the cell from growing and dividing in the face of crisis. And it does this, it inhibits the process of resection, so making that DNA single-stranded. And we were really struck by the fact that when we deleted RAD9, now again, we see this rescue in viability. And so in our wild type strain, no longer is the viability 20%, it goes up to 70%. And we don't see this for our scrambled control. They're equally viable. And in terms of contractions, deletion of RAD9 also rescues that phenotype as well. And so now we don't have 80% contractions anymore. We have something closer to 40 and a return to baseline. And that is really weird and unique And I am teaching cell and molecular biology at Endicott College this semester. And as part of their lab class, we are currently trying to uncover why this is. So Rad9 is attracted to DNA through chromatin modifications on histones. And my students in my lab class are deleting all of the post-translationally modifying proteins that make changes to those histones to see which one of these is really important for this phenotype. And I'm so excited about it. So, kind of in summary, cancer genomes are full of mutational change. DNA repeats are both fragile and they're prone to instability. Single-stranded DNA gaps help exacerbate this fragility and instability, so we get more breaks. And this leads to mutation and loss of genetic information. Single-stranded DNA gaps are found in higher frequencies in cancer cells, and this is particularly true for BRCA-deficient cancers. So to bring it back to our inherited cancer risk. And so this means that it could be an important therapeutic target. And I had some lovely friends write a mini review about this finding that I had found in a Nature Communications paper. And the idea here that Sharon and Catherine and Jenna and I kind of came to is that as you're treated these cancer cells with genotoxic chemotherapy, you have more and more single-stranded DNA gaps. And if you have Rad9, if you're in yeast, or 53BP1, if you're in humans, it will lead to genomic instability and DNA breaks because it will bind near these repetitive regions of the genome, and that will allow for DNA breakage events to happen more frequently. But if we are able to protect our DNA more effectively, then those breaks won't happen, and our cell survival will occur, and that will lead to chemoresistance. With that, I would really like to thank Catherine Freundreich, who is at Tufts University, and Jim Haber, who originally at Brandeis, who gave me this original strain. I want to acknowledge the undergrads that worked on this project with me, Isabella Del Priore, Marjorie De La Rosa Meja, and James Potty. And my funding sources, including the NIH and the American Cancer Society. I would also really love to shout out my mini lab at Endicott College. Many of these students are working on new arms of this work so that we'll understand more about the genetics and molecular mechanisms of CAG and CTG repeat fragility and instability. And with that, I would love to take some questions.
First of all, thank you so much for coming and giving this talk. I know I was very interested for seeing it on the Darwin Fest schedule, so thank you so much for giving us your time. My question is, can you get tested for any kind of like CAG repeats or any kind of repeats? How would that happen? What age can you get tested? What's the process of that?
Yeah, so this is where there's really power in genetic counseling. So unfortunately, I think a lot of clinical availability is limited to family risk. However, I have a son, he's 9, and when I was early stages of pregnancy, I was tested for inherited risk of Huntington's, Fragile X, which is also a repeat expansion disorder, and maybe one other, I can't remember. And so it is sort of part of early screening and pregnancy panels for at least some individuals. And I wasn't particularly high risk, so there's that.
Would I be able to go get tested, or is that something that's happening through for pregnant mothers who are expecting?
I think that's something for a healthcare professional, to be honest.
That's perfectly fine.
Yeah, I'm not that kind of doctor.
Any other questions? If you hold your hand up, I'll run a microphone to you.
I appreciate the time that you've taken to explain everything. I really love the plumbing.
Thanks.
Really. My question is, what is your opinion on the news that Huntington's disease was treated for the first time in a UK trial? And do you think this is the evolution of finding treatment for diseases like Huntington?
Yeah, this was a really, really exciting novel finding. So there's two strategies that have kind of been employed in kind of ameliorating these inherited genetic diseases due to repeat expansions. Option one, which is a favorite of mine, which is CRISPR-Cas9 gene therapy, where we create a break nearby the repeat and we try to contract it that way. And there are steps to be done for that, like there are clinical trials, et cetera. But the one that you're talking about is related to another type of big therapy, which is called siRNA knockdown. And so what the clinicians and researchers are using in that particular method is not to modify the DNA at all, but to modify and degrade the RNA and RNA for the Huntington gene so that you never get this toxic protein. And so both strategies totally work. It's a very different strategy. So it's DNA independent, but it's awesome.
Another question, do you use CRISPR technology while you're doing this study or not?
Yeah, so people in my lab now are trying to see whether or not using CRISPR to induce the double-strand break instead of my special sugar and my special endonuclease, what happens, because what I think we've all kind of come to in the last decade with CRISPR is it's actually a lot harder to effectively make genetic change than maybe we had hoped. And there are a lot of reasons for that, especially in humans. We don't actually like to do homologous recombination all that often, and so that's a barrier to success with therapies and CRISPR. But the other part is it's also just generally ineffective or inefficient. And one reason could be that our human genomes are full of repeats. And if we induce a break with CRISPR-Cas9, hoping to make a targeted genetic change, that there are other genetic changes that are happening. And part of that could be due to these repetitive regions in the genome, which are pretty common.
Okay, thank you.
Yeah.
I think I understood that your point is that recombination can induce all kinds of issues.
Yep.
But isn't recombination a necessary component of creating variation, which is important for evolution?
Yes, and this ties so beautifully into the Darwin of it all, doesn't it? So yes, it does turn out that, genomes are evolving, and a lot of that is recombination mediated. So during meiosis, we have this process called crossing over where you have changes occurring, and that could be mutagenic too. I think the ultimate outcome is, at least in terms of human and animal survival, is do you have a live birth that then survives to procreate? And miscarriages are incredibly common in all kinds of species. And so, yes, we're driving this forward, and we're certainly carrying a lot of these repetitive regions in our genome kind of accidentally, and only in some individuals are they really a problem. But yeah, the genome is ever evolving for sure.
May I ask one more question?
Yeah.
There are also a large number of mobile units.
Yeah.
Do those play into this, or is it a different kind of mechanism?
No, it totally can, because they also use recombination-based mechanisms to move, and that can also lead to mutation and genomic change and be a problem.